All right, we'll get started. Good evening, everyone, and welcome to the Falconer Natural History Lectures from the University at Albany's Atmospheric Sciences Research Center. Tonight's presentation is Blame It on the Moon, Plant and Ecosystem Responses to the August 2017 Solar Eclipse. This is the second Falconer lecture in our spring 2021 series. My name is Mark Boharnwa. I am your meeting host this evening and will be reviewing the lecture protocols and introducing tonight's lecture host. The Zoom protocols for tonight's lecture are as follows. The lecture, all messages sent to the Zoom chat feature, and the post-lecture Q&A are being recorded. Your video and audio has been disabled until the post-lecture Q&A period. However, you may submit comments or questions to the chat at any time. Anything requiring clarification from the speaker will be addressed during the lecture, and any other comments or questions will be addressed during the post-lecture Q&A session. Please do not use the Zoom raise your hand feature until the post-lecture Q&A period. Raised hands will be addressed in the order they were received, and you will be asked to unmute to pose your question. You may also enable your video if you so choose. Please remember that if you're going to ask a question, to ensure that your background noise is at a minimum. Tonight's lecture host is Dr. Christopher Thorncroft, Director of the Atmospheric Sciences Research Center and full professor in the Department of Atmospheric and Environmental Sciences at the University at Albany. Dr. Thorncroft will provide a brief history of the Falconer Lectures, introduce tonight's speaker, and will moderate the post-lecture question and answer period. Director Thorncroft.
Good evening, everyone. I'm just setting up a couple of slides. So welcome to this second lecture in the Falconer series. And before we come to the speaker today, I wanted to say a little bit about the Ray Falconer, whose name is given to the seminar series. So in 1961, Vince Schaefer founded the Atmospheric Sciences Research Center and shortly hired thereafter Ray Falconer as ASRC's very first full-time employee. Ray was often referred to as Mr. Weather, from his early days as a weather observer atop Mount Washington in New Hampshire to his work at the General Electric Research Laboratory in Nobel laureate Irving Langmuir's Project Cirrus. Ray was an enthusiastic disciple of the atmosphere. Ray's boundless energy was poured into public education on weather and the environment. His popular radio weather commentaries, his articles, and his lecture series made him widely known as the voice of the ASRC. Ray initiated this lecture series in the summer of '62 at Whiteface Mountain. And in 1973, ASRC expanded the series to include a spring version on the UAlbany campus, which is this present series. This series is deeply rooted in ASRC's history and offers scientists a unique opportunity to engage with the general public. Indeed, we have a fantastic slate of strong interdisciplinary talks this year, and I would like to thank Dr. Craig Ferguson and his committee for doing such a great job in creating such a strong slate. Before going on to introducing tonight's speaker, I wanted to say a few things about the center for those of you who may be less familiar with what's going on these days. I just want to highlight a few of the areas that we do that are important for the society and the state and the globe. We work on air quality and atmospheric chemistry, observations theory, including many observations that take place at our Whiteface Mountain facility. We do a lot of work on boundary layer meteorology, which is basically looking at the atmosphere close to the surface and how energy, temperature, and moisture are transmitted between the land and the lowest levels of the atmosphere. We also have a growing area in applications of artificial intelligence and machine learning. We've recently deployed some quite strong computing infrastructure to support machine learning through the Excite laboratory, and are contributing to a national institute, a $30 million institute in artificial intelligence and trustworthiness. We do lots of work in climate and weather, including extremes, and we also are very strong in renewable energy, wind and solar, to name but a few. Finally, just want to say that we're very excited about this summer. We are going to be moving into a new building, the so-called ETech building, this summer. And in this building, along with ASRC, we will be joined by the Department of Atmospheric Environmental Sciences, the New York State Mesonet, a Center of Excellence, the Excite Lab, as well as our local National Weather Service forecast office. A dream come true for bringing all of these entities together. And we will also be co-located with the new College of Emergency Preparedness, Homeland Security and Cybersecurity, as well as environmental engineering. A huge opportunity to promote and continue interdisciplinary research. Now, I would now like to pause, and I'll close my slides now and offer Daniel the opportunity to share his screen. And while he's doing that, I will introduce him. So it's a great pleasure to introduce Dr. Daniel Beverly, and I use the word doctor in a celebratory way today because he got his PhD in December. So many congratulations to Daniel about that. So Dr. Daniel Beverly is currently a postdoc at Indiana University in the O'Neill School of Public and Environmental Affairs, and joined this year. He did his PhD work at the University of Wyoming, an expert in botany, ecology, and hydrology. His interests are wide-ranging, including leaf level gas exchange to snow yields at the watershed scale, including coupling of snow distributions and plant hydraulics, and water use within alpine and subalpine ecosystems. He has also studied the 2017 eclipse, specifically to examine the impact of the moon's shadow on an iconic plant species in Wyoming and in the Intermountain West, including the big sagebrush. We are more used to studies of the eclipse, I think, in terms of the environment, or maybe even on animals. So this provides a novel view of the eclipse impacts, and that is the topic of today's lecture. So welcome, and thank you for being here, Daniel. I'm looking forward to your talk.
Yeah, thank you very much for having me. This is very exciting to be here and share some of this work. Can you guys, I can't tell what screen's being shared.
We can see the screen. It looks good.
Okay. So this is some of the work that we kind of ran into when 2017, when the solar eclipse passed over Wyoming. Before I get too deep into this, though, I need to make sure I acknowledge all my mentors and collaborators, all the field techs who've helped with all this work, funding agencies, and the land agencies that have provided this research. So it's a very interesting phenomenon when we think about the solar eclipse. It's well documented. We know when, we know where, we know how long these events are going to last. But these spectacles draw millions regardless of us knowing both when and where all of these events are going to occur. We also know the primary effects in the environment. We think about as the solar eclipse passes, we're going to lose light, reduce radiation, and then eventually it's going to come back. And we also see the moon and sun on a daily basis. There's still something charismatic or romantic about solar eclipses that make them so special. This is one of my favorite quotes that I think about when talking about solar eclipses. This Joe Rayo talking about the 2017 solar eclipse, thinking about solar eclipses as potato chips. As soon as you have one, you want another, and you're looking for more and more. Essentially, you're addicted to the solar eclipse effect. While these rare events also draw millions of people, they have an amazing economic pulse. So if we think about what happened in Wyoming in 2017 alone, we think about 261,000 people traveled to the state. That doesn't seem like a whole lot, but when you think about the population of Wyoming, that's more than half of the whole state, a new influx into the whole population. This also generated over 60 million dollars in less than 3 days. But kind of what we talked about is this charismatic effect. When we think about throughout human history, humans have been obsessed with solar eclipses. We can think about this Norse mythology of Skull and Hati and chasing the moon. And once the moon catches the sun, a solar eclipse occurs. And so we have to think about this great mythology and stories that are passed down through generations. But how many of these people have actually witnessed a solar eclipse? Think about the rarity of these events. And it amazes me at how well these events are documented. Here's a sketch from 1613 illustrating this phenomenon. And we can understand that these events are incredibly well documented through human history, but they are still super rare, and most of the population has not experienced an entire solar eclipse. The other interesting thing that recently is coming around is that the people who have experienced one solar eclipse often have seen more than one, being eclipse chasers. In this little figure, we see these people dancing in 1613. And so this makes me wonder, is there a physiological stimulation associated with the occlusion of the midday sun? To start, we need to see something that why would that influence a physiological stimulation? Something like drugs or salt or sugar, when you consume it, alters your physiology. So if we think about the human circadian rhythm, we can think about this being first originally conceptualized in the 60s, thinking about how phosphate excretion in urine how it follows this really, really strict pattern in humans. And we can disrupt this pattern with different environmental shifts, and we can start changing this with diet and other stimulus. But we also think about plants. Most people don't think about plants as having rhythms or inherent cycles. And so we can actually see this amazingly well, especially since we're allowed to torture plants, unlike any other organism in the world. So take this figure here. We have continuous light hours on the bottom on the x-axis, and we have photosynthesis on the y-axis. We have a mutant or a species that has had the inherent circadian rhythm genes removed, compared to a wild type or a species that has the inherent circadian rhythm genes. And we can see that there's a substantial difference. If we look at the wild type, we can see that there's this really nice oscillation no matter what. So we think about light increasing photosynthesis, and under continuous light, we should continue to go up like the mutant. But there's a physical or a molecular clock controlling these physiological responses. And additionally, we can also think about all of these physiological responses are not the same. So we have stomatal conductance on the y-axis in this figure, where we can see we're starting to disrupt this physiological rhythm in the black with the presence of continuous light. So this allows us to think about, are these rhythms both in plants and animals? So for example, is the emergence of crickets or roosting birds an indication of a rhythm disruption? And we think about animals getting all the attention, but they're really interesting to watch. And we can see this human curiosity, we can apply it to animal systems very well. We can think about cicadas going to roost or crickets chirping as soon as the eclipse happens. We can think about some of the herps migrating or emerging from their dens during the eclipse, or birds roosting. And here's a really good example that came from the 2017 eclipse as well, thinking about invertebrate activity during the occlusion of light. So we can see here on the three faces of the eclipse, and then B activity. And we can see essential as we move from pre-totality to totality and out of totality, we are essentially emulating nighttime or dusk for bees. But animals do maintain all the attention. If we think about all the studies that have occurred from solar eclipses, almost all of this is focused on animals, specifically invertebrates and birds, with mammalias coming in third. But what about these essential life forms that require light? Wouldn't they be interesting, especially if we think about plants? So let's all catch up real quick on general conception of being a plant. So as a plant, my primary objective throughout life would be to grow, accumulate biomass, and reproduce. And all of this is a function of photosynthesis. And photosynthesis is a function of light. And so we really need to think about these fundamental limitations of photosynthesis when we understand plant stress, or the occlusion of light on plant stress. So this conceptual framework really represents where the gas exchange and the carbon and water dynamics of a plant. So just rehashing some of these fundamental biology concepts, thinking about water moving through the plant, through the leaves, and being transpired through the leaves to the atmosphere. But we also have carbon coming in through the same stomata. But the primary concern that we want to think about is sunlight and the chloroplast interactions that are making sugars. And we already have a pretty strong understanding that solar eclipses influence the light quality and the light quantity. So how are these going to influence a plant's life? So if we take a zoomed in picture of where light goes in a leaf, we have all the light coming in. A lot of it gets reflected. Some of it turns into fluorescence. Most of it turns into photosynthesis. So we would assume having a largest proportion being absorbed into photosynthesis, we would see a substantial effect. But we can also test these other fates of light. So here's chlorophyll fluorescence. And so essentially we can see chlorophyll fluorescence is a proxy of how fast electrons can move through the leaf and how fast we can make that electron gradient essential for photosynthesis. So here in these cotton leaves, we can see how stress influences these leaves. So we have 0 days of drought, 2 days of drought, 5 days of drought, and 9 days of drought. And as we progress, we keep losing our photosynthetic capacity as we lose water. And so while this is an extreme case, we might still be able to understand how the fluorescence or the photosynthetic potential is changing even under a rapid shift in light conditions. We can also think about heat dissipation. So this is similar. This is a thermal image of aspen, but we have droughted aspen on the left and moving towards a more well-watered state on the right. And as we see, we have warmer plants on the left, and they're less capable of regulating heat. So more heat is being absorbed compared to the well-watered plants. So now we kind of have this physiological steps that we can evaluate. We can start thinking about which of these are going to be most influenced under the eclipse conditions. So coming back to our question, what about the life forms that photosynthesize? Well, what about them? So let's specifically ask, how much does a solar eclipse influence or reduce photosynthesis? And as kind of stated earlier, it probably depends on what plant you use. So we elected to use the perfect dryland plant. So this is sagebrush. It should be Wyoming state tree, but unfortunately it's not. But it has an amazing physiological and structural properties. It has these little tiny leaves, but they're all clumped and they're always evergreen. But these little tiny leaves and the orientation in this shrub maximizes the canopy exposure to light, meaning that this canopy very rarely experiences any self-shading and maximizes as much as possible of the light interception. So that gives us a good plant who would never see a light-induced stress. Additionally, this plant does really well in the late season. So this eclipse happened in August, and in August in Wyoming, it is incredibly dry, and a lot of the perennial shrubs and grasses have already senesced and seeded. The sagebrush has this incredibly deep rooting system, can go up to meters. And we've done exclusion studies, and we've seen that these plants can go up to 36 months continuous without water just because of these deep rooting networks. So this allowed us to maintain a plant that still had functionality even in the late growing season. Finally, the sagebrush is incredibly common across the western parts of the United States. We think about 16,000 square kilometers from Mexico, but an immense distribution across the western portion of the country. And so for this study, we aimed at focusing up in Wyoming outside of Yellowstone National Park. We also elected to use this site because we had previous instrumentation and previous studies already being conducted at this location. So it's allowed us to rapidly integrate our instruments for measuring photosynthesis while also capturing some of the environmental changes that are driving changes in photosynthesis. And we were able to combine all of these measures with the physiological spot measurements, and then we were able to use the ecological and the environmental drivers to model on a much larger scale. And then the fun happened. We got to sit and wait and watch the solar eclipse. And if you didn't get to experience the solar eclipse, I highly recommend it. So this is just a shadow, but we can see the crescents coming through the trees above us and the shadows being emulated as the eclipse is beginning. So when we think about the environmental responses to the solar eclipse, the first thing we think about is radiation. And so we quantified net radiation throughout the solar eclipse. So across the bottom of this figure should be time, but I guess it got clipped off. And then on the y-axis, we have net radiation. Now we can see we actually have a more negative net radiation flux relative to first thing in the morning. And we can see we had essentially a perfectly clear, almost no cloud in the sky day with this trace throughout the solar eclipse. The gray represents the partial eclipse, and the dotted line represents the 2 minutes and 18-second total solar eclipse. And so with temperature, we were also quantifying temperature with the net radiation, as it's another primary driver of photosynthesis. So as we would expect, we increased temperature throughout the day. Once again, sorry, this should be time across the bottom axis and temperature on the Y. But the story begins with, as we move into the partial solar eclipse, we think about the decreasing temperature. We took a little lag, and then following totality, or the dashed line here, we can see that there remained a lag. So net radiation did not have a lag. It hit zero and then rebounded immediately, which makes sense. But the temperature and the cooling effect to that long wave radiation coming out still affected the air temperature until ultimately recovering after the partial eclipse. So while the environmental drivers are very essential to understanding the effects, it still has not answered how much does a total solar eclipse reduce photosynthesis. So to measure photosynthesis, we used what we call an infrared gas analyzer, which clamps onto the leaves and can track the solar radiation in the environment while quantifying the gas exchange at the leaf. So on this image or this figure here, we think about time on the x-axis and then our net photosynthesis. And our net photosynthesis is represented by this green squiggle line. And we think about, we increase and we have this noise, right? So this is every time it comes back down to being less noise is the instrument self-calibrating. And so these gas analyzers are pretty noisy, but we still get this really nice trend in our daily photosynthetic rates if we follow the green line. As we can see, we are going below zero just before totality or the dashed line, and then recovering back. So we're respiring as we're below this solid line at zero and eventually recovering. Well, this temporal variation was not that substantial. So we lost 2 hours of our maximal photosynthetic rate for the day. We also think about how did the plant influence heat dissipation. So we measured heat dissipation with 2 different instruments. We have the same instrument that you see here, plus a handheld instrument that we use to validate it. And we're saying this nice decrease, but we also did not see the lag in heat dissipation as we expected with the radiation and temperature. And also the heat dissipation, so we never reached zero. So the plant never really acclimated to a dark or a nighttime status that we would have expected with zero photosynthesis occurring. But we still also need to think about how does the occlusion influence the sun? So we talked about the sagebrush never seeing any shading or light stress. And can we actually address this occlusion of the sun emulating this dusk response or influencing the biological rhythms? So if we think about this figure, we have light flux on the x-axis. So as we have pre-eclipse conditions around 1,000, and then we have chlorophyll fluorescence. And we think about chlorophyll fluorescence is recovering or moving higher in value as we decrease in light. And so this is our measured response as we moved into the darkness or into the solar eclipse. And we expected this nice transition to follow the same linear path coming out of the solar eclipse. But as soon as the eclipse happened, we saw this major shift in fluorescence capability at the leaf level. This major drop immediately following the reemergence of light. I mean, so we're still at a very, very low light level here, but this substantially decreased the fluorescence. And we did not recover this fluorescence response until we reached about 1200, 1500 at the light flux density. So we can infer that this major drop or this just drastic shift from our linear trajectory to being a stress response that we detected in this fluorescence parameter. And then, so the eclipse seems to have disrupted our daily cycle. And so we think about how much did that disrupt our cycle? We think about scaling out across the big picture. So we think about how many acres of sagebrush there are. But we need to think about how to do this efficiently. Can we use the previous day? We were there the previous day. but the previous day had a bunch of clouds. So that might not be as representative of the total carbon loss for that entire day. So our next simulation was to use the eclipse response as our light response and model light flux densities based off, or model photosynthesis based off of light flux densities. So we think we get this nice response with light flux density on the x-axis and photosynthesis on the y-axis. And as we move through the eclipse, we start coming down in this nonlinear response, thinking decreasing in photosynthesis until we reach 0 light and a negative photosynthetic value. And conversely, we also get a progressive change in recovery back to full light conditions. And so we still have this noisy response, as you saw with those noisy calibration fluctuations that we saw. But this light response allows us to model the light status across the entire country and then project that disruption across the entire Western United States. So this is some of the eclipse footage. This is just a still image, this is eclipse footage that was captured from a drone. And one of the cool parts about this is essentially watching the sunset in the east. So this drone capture was done by my dad, who launched a drone a couple minutes prior to the totality, and we were able to capture this. But we think about across this entire path, we think about all these little polygons, or this totality zone in yellow, and how much carbon was lost across only that. We think about a no eclipse day versus an eclipse day based off of that light response. And so we think about a 14% decrease in total carbon uptake from the eclipse versus a non-eclipse. And so as the title of my talk was originally called, Blame It on the Moon, we originally proposed this as saying, can we blame climate change or the increase in atmospheric CO2 concentrations on global eclipses around the world? And from our back of the envelope calculations based off of this 14% decline, we essentially would need 24 million eclipses per day to reach the current CO2 concentrations from pre-industrial revolution to now. So blaming climate change and the large pulse of the efflux of carbon to the atmosphere is probably not consequence of solar eclipses. And so we also think about how much carbon is this, 40 gigagrams versus 30 gigagrams. This seems like a lot, but when we really think about how much carbon was fluxed from just carbon emissions from those 200,000 tourists coming to Wyoming, was more than double this, estimated at 81 gigagrams. So this hopefully starts to be able to fill in some of these questions and some of this pie chart with how plants are also being drastically impacted by the solar eclipse. And so we were able to show that plants and other photosynthetic life forms are drastically influenced by the occlusion of the sun. And we also interestingly looked at how biorhythms or circadian clocks are also impacted by the sun. So another exciting kind of component that's actually closer than I've ever thought is the next solar eclipse that's going to cover through much of the North America and the United States. So I think about this 2024 solar eclipse, and here I can have my highlighted where I did this 2017 work thinking about Wyoming. My current position is in Bloomington, Indiana, which is perfectly aligned with this next solar eclipse conveniently. And then up near Albany, you guys will be extremely close to the 2024 solar eclipse. The only caveat is this eclipse is in April. So I foresee the plants being not as photosynthetically active across this northern range of the eclipse. So we might have to go to Mexico or Texas to actually quantify some of these responses. So the quick take-home points for this little talk was thinking about there was a 14% reduction in carbon uptake by sagebrush communities. And then we also had unusual light changes in stress, stresses out photosynthetic machinery, and disrupts daily biological rhythms. But I really want to leave with this question. Are solar eclipses truly addictive due to the physiological disruption of the circadian rhythm? Because I know after the solar eclipse I saw in 2017, I ultimately wanted to see another one immediately. And so with that, I'll take any questions. So questions in chat. Does the plant response to this eclipse provide any new insight in plant response daytime? Yeah, so that's one interesting question, is thinking about this cloud cover comparison. I mean, so just from the light response, clouds, even the darkest thunderclouds still transmit light. And so I think replicating this would provide more insight into actually understanding, but I think we really want to go down to the molecular level and start looking at some of the DNA, because we can measure the DNA fluxes at approximately the same rate, looking at RNA sequencing and DNA sequencing patterns as the eclipse is progressing as well. So the second question is, there's a 14% decrease in carbon uptake the day of the eclipse, presumably with concurrent savings transpiration. So with the water limited environment, Wyoming sagebrush, do you think the seasonal carbon take would be affected at all? Yeah, absolutely. I think it would be affected. The response and how fast the plants responded to the loss of sunlight is probably a response to the reduced light availability. And so that's absolutely, I think, say if that happened in Missouri or some of these less water limited ecosystems, the effect, the 14% would not be as drastic. But that 14% is incredibly conservative, considering that the penumbra or the partial solar eclipse was not accounted for. And so we know that there was a reduction in the photosynthetic capacity even in the partial solar eclipse that we weren't able to account for. So yeah, is there a difference in clouds and eclipses? Absolutely. The light levels. There's also some major differences in the light quality. Thinking about this fluorescence dynamic, we have a big difference in the light quality, being the reds and the blues are transmitting at different rates. With different plants have changed differently than sagebrush. Yeah, I mean, definitely. Some plants, sagebrush is, I mean, was already extremely water stressed. And so some plants might not have even photosynthesized if they didn't have the deep enough roots.
All right. All right. Does anyone else have any more questions for Daniel? You can raise your hand, or you can put it in the chat. Chris Walchek had a second follow-up question to his original question. Is there a lag in the photosynthesis response?
There was, so that's why that temperature dynamic, it wasn't the same 9-minute lag, but it was in between the 0 lag of radiation and the 9-minute lag of temperature. So there was definitely some coordination in the environment with the temperature change and the light.
Can I ask a question? Can you reproduce this in the laboratory? Can you do any of this in a lab environment?
We can do some shading work, absolutely. Recreating the light conditions. So there's a bunch of the atmospheric scientists were quantifying the variation in light quality. That's what we can't replicate.
Right.
So the light level, yes, but the light quality, no. So how would we improve the instrumental suite? Partially doing some of the genetic assessment, as well as so for the 2024 Eclipse, we'll also be running sap flow and eddy covariance. They'll be going over the Morgan Moreau Ameriflux site here in Indiana. So we'll have a lot more data to be playing with at that one site.
So you're not coming to Buffalo?
Probably not in April.
I think
Yep.
Stephen, is raising his hand. Stephen Riggles. Let me see. I can unmute him. Stephen, can you?
Unmute. Okay.
Yep, there we go.
Would the performance of solar panels be similar to the reduction in carbon uptake?
Sure, I would assume potentially, yeah. I don't know. That's a good question. I'd have to. I'm not completely versed in the solar panel mechanism world.
Any other questions?
Looks like Lucy had one second follow-up question in the chat. How does the short day impact the normal biorhythms over time?
Well, yeah. So the short days, your body and even plants can be. You can have a longer clock. So like most molecular symptoms are like, so we think about phenology. So there's like an annual clock and then a daily clock. And so the sun and environment often syncs those clocks, but the solar eclipse is just a novel disruption. And so we can see disruptions of clocks and biorhythms with other stress events. Like very commonly, we can disrupt it with drought. I mean, that's how we really test this in the growth chambers, is we can drought plants and watch them, or we can give them 24 hours of light.
And there's another question there. Is there any opportunity for citizen science in 2024?
Absolutely. I mean, there's going to be hopefully a huge campaign. I was amazed at how much interest was around the 2017 eclipse, and I'm hoping the 2024 one is even bigger. Like I think that in Wyoming there was a bunch of work. People were collecting. The university was sending out sample kits and all sorts of things. The Indian reservations got involved heavily with citizen science and doing observations of animals and all sorts of things.
Did you notice any animal behavior where you were sitting?
Yes, you could definitely tell, like birds roosting.
Mm-hmm. So Craig asked a question. Other than tourism, what are other ways people monetize eclipse events?
Well, I mean, there was a lot of like swag to like the post office. I was a victim of buying stamps to commemorate the solar eclipse. So, like, yeah, just knickknacks and stuff.
So what does Mark say? Is there an eclipse anonymous for those addicted to solar eclipses? Asking for a friend.
Sure, I don't know. I mean, some of these people have seen like 30 of them, which
Mhm.
is really amazing to me.
So is it, I mean, it sounds quite novel, the work you're doing with the plants, as compared to, say, with a lot of work that's been done with animals and stuff. I mean, is it been much less done on plants than animals?
Absolutely. I mean, so Kathy Stepp and a few other people have looked at sap flow. And so if temperature decreases, your transpiration is going to decrease, and so is photosynthesis.
Mm.
The more novel aspect of this work is looking at the fluorescence and some of the circadian stress,
Mhm.
rather than just the expected physiological responses from changing light.
And I'm guessing a plant will never experience this stress any other way, right? I mean,
No.
you can't go from cloudy to uncloudy so rapidly.
Yeah, I mean, you can't go from essentially, I mean, so we are also midday peak sunlight, so maximum light flux. So you can't really go from that to darkness back to maximum light flux in that fast.
Any other questions? Dustin, I mean, I can't see hands going up because I can't see any other people, but I guess you can, Dustin, right?
Yeah, there are no other hands up, so we are good on that front.
Okay. So I think Mark's doing an announcement here that to everyone that the recordings, including last week's as well as this week's, are going to go up on a new YouTube channel for SRC. So it'll be good to catch up with all of these talks, and later ones, if you can't make them, they'll be there as well for you to enjoy. Craig, seems like a potential geoengineering angle to this work.
Yeah, so how much does cloud cover reduce carbon uptake?
No, the first question is, I think, Craig, do you want to explain
Oh.
that question?
Hi, Daniel. I was just thinking in terms of, the controversial idea of geoengineering. So kind of placing, reflectors in space, et cetera. I think that might be the closest we get to the eclipse effect, right?
Oh, yeah. Yeah, I guess that's true. Yeah, I mean, the hardest thing, like, is the light. And it's pretty amazing. Like a couple of the papers that I've seen actually looking at the light quality, it's pretty astonishing to me how different it is. And when you actually saw the eclipse, everything was blue. It was really, really amazing. I've never seen anything like that in my life.
And then the question you just referred to by Lucy Comley, how much does cloud cover reduce carbon uptake?
I mean, it depends. But like, so some of the best work that we've seen on that is we could see like a 80% reduction under major thunderclouds, like in the Kansas grasslands, is some of the best work that's really tried to understand some of those major thunderstorm events. But so that's kind of a double-edged sword to think about. So usually those are accompanied by rain. So you have longer photosynthetic uptake later in the season. So
We do have a late hand that came up here. Joanne has a question. Joanne?
I'm sorry if you already went over this, but I'm curious to know if you've come across any studies that indicate the moon or solar eclipses have impact on human beings.
I haven't. That's why I kind of wanted to know if this addiction behavior is a clock response or some sort of dopamine response in the human physiology realm. But I have not seen any work on that.
Any more questions? No? Okay. Well, I think we should draw that to the meeting to a close. I'd like to thank you very much, Danielle, for intriguing, interesting presentation and talk, and love to have you come and visit us in person one day.
Yeah, that'd be so. Yeah, that'd be great.
Buffalo 2024, maybe.
Yeah.
But thank you very much, and look forward to seeing everybody again next week. Okay, thank you.
Awesome.